Bruce Alberts refused to introduce me, and so I thought I would take the next half an hour, 45 minutes to go back over my background. And but actually, my name is Marc Kirschner. I was on the faculty here at UCSF for 15 years. After that, I went into obscurity, but I'd like to. I'm at Harvard, which medical school, which also probably I should introduce. But anyway, nevertheless, during the 15 years I was here, it was definitely the most exciting and in many ways the most rewarding part of my life. And coming back here is a real treat. And being able to say some things about Mike Bishop, who is both what, pardon? Yeah, you know, it's this is Mike. This is my chance to talk. You know, you get your chance, but not publicly, fortunately. But anyway, nevertheless, in any case, I thought what I would do is everybody's taking something about Mike's public career, and I will refer to that, but I want to talk about Mike a little bit as a person, because it very much, apart from everything else, also impacts on the tremendous contributions he's made as a scientist and as a leader in this institution. And I want to give some context for what they were. So, first of all, I think to some people Mike presents a very formal portrait, lots of complex language and allusions to things that nobody ever experienced except he did, except for him. But in fact, he's a terrific human being, very passionate about everything, passionate about sublime things, passionate about things that aren't so sublime. And he's thoughtful about virtually everything. And I tried to look for a picture of Mike deep in thought. And so I found one, just to give you an idea. And actually, this is good because it has a sort of control here. I was also deep in thought, but I think you can see quite obviously that my thoughts are not as deep as Mike's thoughts. It's immediately obvious. All right. Anyway, so let me say some things again about him. He's a terrific friend, as many of us here know. But he's also been passionate about helping people at every stage in their careers. In the many people in his lab who've gone on and done wonderful things attest to that, and they're not up here speaking, but I can speak to that. I also can speak to many faculty here at UCSF, and I don't want to give specific examples, it's sort of embarrassing, but who really were helped by Mike achieve the kind of credit and recognition and opportunities that they couldn't achieve without his help. And I think that that's certainly a major part of his contribution that doesn't show up in his CV. Also, Mike could be counted on to help on almost any good cause. It has to be a good cause. Better it's a cause where he's not being publicly praised for it, where it's an unmet need in the community. And I saw that in many opportunities, and we could spend the rest of the morning sort of listing those things, and I don't want to, but my own experience with him came with the tremendous contributions he made in public policy to also to the American Society for Cell Biology, and to establishing the caucus and working in the House of Representatives on biomedical science. And all these things were done without any obvious credit given, and they made all the difference because he brought that same concentration that you saw on that first slide to everything that he does. And all of us who've worked with him on these things are just grateful for that. Now, we often, Mike and I and others here at UCSF, joined in serious undertakings, and I have a photo of that too. I think this is a picture that where we were, the four of us were writing something for Science magazine in the days when they used to publish interesting things. And you can see Mike. Well, he's the center of this group of the four of us. You might recognize. So it was definitely a collaboration. I think, as I remember, Bruce inspired it. I talked about doing it. Harold wrote it, and I think Mike typed it up. But nevertheless, nevertheless, it was a collaboration, and we all got equal credit. Anyways, I also, and this I want to be serious about, because he doesn't, but many people don't, did not experience this in the scientific world. But he, I think a tremendous part of his strength is his family and his wife, and I just wanted to show a picture of that because I think they're a tremendous team, and Catherine is definitely someone who's challenges Mike and keeps him honest and provides tremendous amount of support and is, as I say, is a wonderful critic, and I think it's in thinking about what a wonderful person Mike is and how much he's contributed. I think one has to think about Catherine and the two boys, but also people I really haven't met. I must think that Mike's parents, he might cringe about explaining his greatness by his religious upbringing, but somehow they must have given him some set of values and standards that he carries through his life, and so I think all of this background does a lot to sort of explain the foreground that most of us see. Now, I can't really explain his obsessive appetite for knowledge in every subject. He's the most obsessive reader I know. Books they were hardly contained in the large chancellor's house. I think they're now a serious problem for where he lives now. They just basically take up more space than anything else, and he actually reads them, which is, I think, interesting. And through this, he has just an amazing understanding of history, of science, of biography, of novels, of art, of sports. It's just to be wondered at. And he's just got a wealth of knowledge from baseball to zoology, from infectious disease to classical music, from everything. He's a collector of art. He's an avid hiker. He's an accomplished fisherman. In fact, and there's a sort of informal side of Mike. I mean, if you see him in Aspen, Colorado, or even walking on the Tennessee Valley trails, it's where he, in a way, seems most comfortable. And I think this is probably not a view of Mike that many of you see often, but it's one which is, I think, very much of a part of his of what he loves about life, and I think again it somehow all comes together. Now, I want to just add the final thing, which is the most obvious thing. Mike is a lover and a connoisseur of the English language. This has been crudely ridiculed in the past by UCSF graduate students in a famous skit where Mike speaks incomprehensibly in polysyllabic words, while an interpreter translates this into an insipid monologue. And this has to be wrong. I mean, first of all, what sense is that for the standard of comprehensibility to be a UCSF graduate student, or even a Harvard graduate student? Because I mean, it's to our students, both Sarah Palin and Ludwig Wittgenstein are both incomprehensible. And so, Mike somehow fits somewhere in. And surely the reasons for Mike's incomprehensibility must be different from Sarah Palin's, or even Ludwig Wittgenstein's. So I leave it for you to decide if Mike is where he stands on this pantheon of incomprehensibility. But I think we should give him the benefit of the doubt, especially if we can't understand him. All right. In any case, I just want to say that I've learned a tremendous amount from Mike about culture, and I also, like I said, one more slide here, just showing again Mike in one of his pensive moods and thoughtful moods. This was Mike when he embraced the frog, which was a short-lived thing. He went back to other organisms. But in any case, but I would want to say that I've learned a tremendous amount from Mike about culture and art and science, even a few scattered vocabulary words. But what I learned most about was to value the contribution of others, and I hope to hold myself to a higher standard as a result of knowing him, not only in science, but in the culture that makes science possible. Now, it's not likely any of us will come close to Mike in his accomplishments, but I think every one of us can benefit from his example, and we should all be grateful for that. I thought I would end with a little bit of a quote, which was a famous one everyone knows, that Alexander Pope said that the proper study of mankind is man, and this leads me into, I think, the introduction of the talks, and that is, he didn't mean by that we shouldn't be studying Rous sarcoma virus or Bacillus subtilis. I mean, and I think, in fact, the focus of that essay on man was to favor science over studying religion. And I think, and I consider Mike, and that's on the scientific vein, and as we enter, as I get into what we're going to be talking about today, is not as a cancer scientist or someone who's doing translational medicine, although he's doing a lot of that now, but as someone who values and epitomizes basic science, because after all, nothing, is more unrepresentative than Rous sarcoma virus or chickens or whatever he studied. This was not human biology. And yet what came out of those understandings in Mike and Harold's lab were insights that really transformed the study of human biology. And so I think what I think about what UCSF stands for in my mind, and the time I was here, and the example that Mike and Harold and others have shown, is that they were really appreciative of fundamental discoveries. They did them themselves. They thought they were the way into science. They understood model systems as the important thing to do. And they were willing to project those into human biology. And I think the statements that are made often glib today about we can just make human beings the next model organism just don't make any sense to me, and they don't make sense in terms of the career that Mike had. And yet, that people are sort of seem to rewrite history all the time, and that's not how it was. I mean, Mike, I think when he really found in himself his inner cell biologist, and that made all the difference. So today, the subject of this discussion is decision making in cellular decision making, and in a way that's a kind of a metaphor for almost all the important problems that exist in biology today, and therefore it's not possible, I think, to have as comprehensive a view of that question as we heard in the last wonderful session on malaria, where you could at least, we knew there, we're studying here, it could be anything. And yet there are some principles there that have emerged from many things that people have done. And it's sort of interesting in thinking about who would best exemplify an understanding of decision-making that's important for human biology, important for all of biology. There aren't very many people who've made the contributions at this scale of Rich Losick, whose contributions, in fact, have not even been in multicellular organisms or even in eukaryotic organisms, but have been in prokaryotic organisms, which is what he's worked on, as far as I know, almost completely in his entire career. And yet, in these simpler systems, these genetically modifiable systems, these physiologically understandable systems, come the basic insights that all of us need to understand in biology. And so I just want to welcome Rich. He's a professor of biology at Harvard College. As he asked the question last time, he's extremely interested in undergraduate education. He's a member of the Howard Hughes Medical Institute. He received his bachelor's degree in chemistry at Princeton University, his PhD in biochemistry at MIT. He was a junior fellow at Harvard, and he's a recipient of many awards, but a member of the National Academy, American Academy of Arts and Sciences, member of the American Philosophical Society, fellow of the AAAS and all those things. But what he has been has been the strongest, I think, intellectual force I know of in really dealing with the question in the modern age, and after Monod and Jacob, of how decisions are made by cells, how they integrate extracellular signals, how they transform their own environment. And his talk is entitled Decision Making in a Microbial Community. Rich.
Mark, thank you. The problem with such a glowing introduction is trying to live up to it. Let me say as an outsider that obviously I'm honored to have been asked by Mark and Bruce to participate in this symposium in honor of Mike. But as an outsider, let me add that under Mike's leadership, UCSF has created this unique culture that values teaching and scholarship, and that's been widely influential across the country, even at my own institution, which is sometimes recalcitrant to change and sometimes needs emissaries to come directly across the country to help fix things. I'm going to talk about decision-making in a bacterial community. So traditionally, bacteria were seen as solitary creatures that went about their business on an individual basis. But increasingly, we recognize that many bacteria, if not most, are capable of living in complex multicellular communities called biofilms. These biofilms are of great significance and importance in medicine, in industry, and in agriculture. Cystic fibrosis involves biofilm formation in the lung by Pseudomonas aeruginosa, endocarditis, Staph aureus, indwelling devices, surgical implants, and catheters are at risk for biofilm formation by Staph epidermidis. Dental caries, strep mutans, the inside of pipes, water pipes undergo corrosion due to biofilms, the hulls of ships. So this is something of wide significance, but also, and this is what I'll be emphasizing, of great interest from a point of view of basic biology. The particular biofilm-forming bacterium that I'll be talking about, which has already been introduced, is Bacillus subtilis, and it makes this absolutely beautiful biofilm with the interesting architecture, and someday I would love to be able to understand in detail how this architecture arises. Now, I'm just back from a substantial trip to China, where I was overwhelmed to realize that almost every province of China has an agricultural university where there is a microbiology group that's isolating Bacillus subtilis and its relatives from the soil as plant protectants. I even got to visit in the far northwestern region of China, in the Xinjiang province, in the town of Urumqi, a factory that receives these strains from these academic researchers and then grows up vast amounts of bacillus spores, which it then distributes to farmers who include them with seeds. And so the spores germinate, and they then colonize the roots of the plants by making biofilms, which protect against a variety of microbial pathogens. In the United States, we largely use chemicals. In China, they're cleverly using microbiology. So I want to address two questions about biofilm formation in Bacillus subtilis. First, how do cells decide to create a biofilm? And then how do cells decide to leave the biofilm? Because as you'll see, biofilms have a lifetime. They assemble and then they necessarily disassemble. At the heart of what I'll say about this decision-making is a self-reinforcing deterministic switch that I hope to lead you through as clearly as possible. But then I'm going to make a little digression and, for a moment to tell you that the same circuitry that's responsible for this deterministic switch in another biological circumstance is the basis for a noise-driven switch. How do cells decide to leave the biofilm? And here the main point will be the discovery of a small molecule factor that appears late in the life cycle of the community and tells the cells to disperse. Okay, so here's a closer look of cells in the biofilm, and there are two key features I want to draw your attention to. First of all, the cells are in long chains that are linked together end by end. Secondly, the chains are parallel and they're cemented together by an extracellular matrix. I want to talk both how the matrix is made and then about this chaining process. So the matrix consists of two components, a polysaccharide and something that's a fiber that's amyloid-like that'll become important later in my talk. So am I blocking everybody over here? Is that right? The polysaccharide genes and the amyloid fiber genes are initially in growing cells, we know biofilm was being made, held silent by a repressor R. When the cells receive environmental signals, various kinds of stresses that trigger entry into the biofilm state, that leads to the synthesis of an anti-repressor called I. I binds to R, antagonizing it, inhibiting it, thereby leading to derepression of these matrix genes. Notice that I've color-coded this with red referring to control at the level of gene transcription and blue at the level of protein-protein interaction. So I'm going to lead up to something that's a little bit subtle, and this color coding will be an important guide as I go along. What about chaining? So chaining is controlled by a family of genes called autolysins. When bacterial cells, when B. subtilis complete cytokinesis, there remains after cytokinesis some cell wall from the division septum that needs to be digested by autolysins so that the daughter cells can separate. If autolysin genes are on, then we have lots of individual cells. If the autolysin genes are off, then the daughter cells remain linked together in long chains. What happens during biofilm formation? Well, during biofilm formation, the autolysin genes are repressed. And that repression requires an additional regulatory protein called SLR, which is itself under the negative control of R. So when I antagonizes R, not only does that lead to derepression of matrix genes, but also of the gene for SLR, so that the SLR protein accumulates. But SLR alone is not capable of repressing autolysin genes. Instead, it captures R to make in SLR complex, and it's this heteromeric complex that's the proximal repressor of autolysine genes. So this is what I want to focus on now, because this has interesting implications. Notice then that SLR is repurposing R from being a repressor of these genes, including the gene for itself, to being a repressor of those genes. So here I've just drawn that simple linear relationship. SLR captures R such that R can no longer repress the gene for SLR, and instead it represses autolysine genes. It's part of the complex. So its specificity has switched. This means, in effect, that SLR is inhibiting R by titration, if you will, by sequestering it in a complex. It follows from this that SLR and R are mutually antagonistic. And so if I've been clear so far, nothing up my sleeve, I'm just going to recast what I said in a form of a simple feedback loop, in a simple double negative loop. So R represses SLR. It's an antagonist of the gene for SLR. And meanwhile, SLR is an antagonist of R by titrating the protein in this complex. This arm acts at the level of transcription, and this arm acts at the level of protein-protein interaction. So this is reminiscent of a classic example of a self-reinforcing double negative loop, that of phage lambda. So phage lambda, at the heart, there's a switch that involves two repressors. Each of these repressors represses the gene for the other repressor. In this case, both arms operate at the level of transcription, and as I've been saying, the biofilm switch has the additional feature that this protein R gets repurposed to have a new specificity. But the logic is similar, and switches of these types have two states. So initially, in a growing cell, we can say that the switch is in an SLR low state. That is, R is repressing the gene for SLR. Since there's little SLR protein, R is not being inhibited, and matrix operons are off. So that's how cells exist when they're growing. Now, environmental signals trigger the synthesis of the anti-repressor I. That antagonizes R. That leads to derepression of SLR. SLR titrates, inhibits R, leading to yet more SLR synthesis, and we get the self-reinforcing loop. As a consequence, autolysine genes are now shut off, and matrix genes are now turned on. If this is right, and if this is a switch that can exist in two stable states, then it predicts that it should have a property called hysteresis, or just simply memory. That is, if we just through a pulse of making SLR protein artificially drive it into the SLR high state, the cells should remember that they're in their state even after the initiating signal is removed for some generations. So here's such an experiment. Here's the circuitry that I've been describing. And in this experiment, all of these genes are present intact in their wild-type configuration. But in addition, we've introduced an additional copy of the SLR gene that's under the control of an inducible promoter, which we can switch on or off by adding an IPTG inducer. So without IPTG, most of the cells are individual. If we add IPTG, we can drive all of the cells into chains. And now we wash away the inducer. Will the cells remember that they're in this high state? And as you can see, at 2 hours and 3 hours, even out to 5 hours, the cells stay in this chaining state. They remember, even though the original signal is gone. As a control, we can do the same experiment, but in cells that are mutant in the gene for SLR. So we have single cells, we drive them successfully into chains with inducer, we wash away the inducer, and they have Alzheimer's, they forget, and they quickly go back to being individual cells. So The point of all this is that this is a device for cells to stably stay in a state of promoting multicellularity. And I think this makes good sense. This is a community affair, and if the cells are going to form this elaborate community, it's important that everybody do so and in a persistent fashion. Okay, so if permit me, I'm going to digress away from biofilms for just a moment because there's something else nifty about this switch. This is the deterministic switch in the sense that the cells are driven from one state to the other state by this I protein, which is made in response to environmental signals. Turns out that the same circuitry is deployed in a different biological context for something that is akin to what we call bedhedging, as I'll explain. So the same circuit acts as a noise-driven bistable switch for bedhedging during growth. Turns out that when B. subtilis is growing exponentially, lots of nutrients, steady state, it isn't a homogeneous population of cells. It's actually a mixture of two kinds of cells. The chains that you've already seen, shown here in red, and swimming cells, shown here in green. So these are motile cells, and these are sessile cells, these long chains. And the way we think about this is that B. subtilis is hedging its bets. It doesn't know what's in store for the future. So the long chains stay put and exploit the niche that they're in. But these motile cells are nomads that swim off looking for new sources of food. And the final point is that these cells switch between these two states stochastically. There's little I present in growing cells, and so this is a noise-driven process. So the same switch can act in a controlled fashion, in a stochastic fashion, and this is actually a big theme in biology, is beyond this one nice example. Okay, so I've told you about a deterministic self-reinforcing switch that's at the heart of cells entering this state of multicellularity. So this is how the cells decide to build a community. But they can't stay in that community forever because nutrients become limiting, waste products accumulate, and so cells need to exit the biofilm and return to an individual existence. So I want to tell you now about the discovery of a small molecule factor that triggers biofilm disassembly. And there are three questions I'll address. First of all, the most obvious, what is it? Second, how does it work? And the most provocative part is could it be universal or at least widespread? And is there even a chance that maybe it could be useful? So here's the phenomenon. This is at 3 days. This is a robust bacillus biofilm. If we wait several more days, the biofilm disappears. You can see an orange pigment that's generated during biofilm formation. The cells haven't disappeared. They've just dispersed back into solution, and they're no longer at the surface in this complex architecture. So we reasoned that as the biofilm ages, at some point it produces a factor that's triggering this disassembly process. And if so, we should be able to recover it from the conditioned medium and add it to a fresh inoculation and see if it would block biofilm formation. So that's what this experiment shows. We took this conditioned medium and added it to a fresh inoculation. And if you compare this image with that image, you can see that it's had a profound effect in blocking biofilm formation. What is it? Well, to make a long story short, it's 4 D-amino acids: D-tyrosine, D-leucine, D-methionine, and D-tryptophan, with D-tyrosine being the most potent. But most potent of all is the combination of all 4, which acts at the low nanomolar level. So in biology, biology is dominated, of course, by the L stereochemistry, and all ribosomal generated proteins have L amino acids. But there are a few places in nature where the mirror image also exists. And I think the way to think about this is that nature has reserved the D form of amino acids for other special tasks outside of being parts of ribosome-generated proteins. And this is another example of that. So let me first demonstrate that we can substitute the factor for with the D amino acids. So here's untreated, and if we have a culture to which we've added D-tyrosine, no biofilm gets made. If, as a control, we add L-tyrosine, nothing happens. The biofilm gets made normally. If we do the same with D-leucine, we can block, but we don't block with L-leucine. We can even take a preformed biofilm and put a droplet of D amino acids on it, and within a few hours it begins to break down, as shown here. So a droplet was put on the biofilm, and a few hours later it's starting to break down, and as a control, if we put a droplet of L-tyrosine, nothing happens. Okay, so where do these D amino acids come from? So D amino acids are generated by a family of enzymes known as RASmases that take the corresponding L form of the amino acid and convert it to the D form, the mirror image. And once again, to make a long story short, we found two genes in the chromosome that are partially redundant with each other, but if we knock them both out, they block D amino acid production. So D amino acid production is governed by two RASmase genes, RACX and YLME, that are switched on late in the biofilm life cycle. So these genes are actually silent until several days into the history of the biofilm. Then they get turned on, and then the RASmases, then the D amino acids start to appear. And knowing all this, we could do a nice experiment. We could take one of these RASmase genes and put it under the control, once again, of our inducible promoter. And without inducer, we make a biofilm just fine. But if we add inducer at the very beginning so that the RASmase is being switched on prematurely, then we never make the biofilm. Okay, so how does the factor work? That was my second question on this part. What's it doing? This electron micrograph shows you these amyloid-like fibers. So sometimes they bundle into these thicker structures, as you can see. So they're quite conspicuous in the biofilm. And if we blow this up, it looks like that the fibers are emanating from the outer surface of the cells, as if they're anchored there. So bacterial cells are surrounded by a huge macromolecule, cell wall known as the peptidoglycan, that gives the cell its shape and rigidity. And it's known that cell wall is one of the places in nature where D amino acids are found. Not the four I told you about, but other D amino acids. And so the hypothesis was that these D amino acids are being incorporated into the wall and thereby triggering the cells to jettison these fibers. That maybe the fibers are anchored in the wall, and when the wall gets modified by incorporation of D amino acid, the fibers get released from the cells. So we did the experiment. That's the hypothesis. Incorporation into the peptidoglycan triggers release. And here's an example of an experiment in which these are D amino acid treated cells and have few, if any, fibers emanating from them. We can still find fibers, but they're not attached to cells. These little dots are gold particles from the immunostaining against the protein, the principal protein component of these fibers. Okay, so I've said what it is, what it's doing, and now finally I want to come to the part that has us excited, but I've been around the block long enough to know that most things that seem promising medically turn out not to be useful in the end. So right now we're in this window of time when this sounds like it might be something useful. So it turns out that these D amino acids are produced by a wide range of bacteria. As I haven't properly credited Matt Waldor, whose earlier work was very influential in what we did. So a wide variety of bacteria besides B. subtilis make the amino acids. So could it be that other kinds of bacteria also control disassembly of biofilms in a similar manner? I guess I should add that the way it's as if multicellularity, in keeping with a question Mark was asking me at dinner, arose independently multiple times in the bacterial world, and the mechanisms can often be quite different. But maybe this feature of biofilm control is common to many bacteria. So we attempted to test that idea. So I'm going to show you examples with Staph aureus and Pseudomonas aeruginosa. And we look at biofilms in a different way with these bacteria. The bacteria stick to plastic when they make a biofilm, and so we can wash away bacteria that have not adhered to the plastic.
Basically, I fell in love with UCSF when I was considering my career options, when I realized the big commitment of the institution to bringing forward a quantitative thinking into biological questions, and the determination to bring multidisciplinary research to the heart of biology. And this has many implications in this day, in this age of large amounts of data and our realization of how complex biology is. The implications of that are that we are now more and more interested in deciphering generalizable principles from this data and understanding the underpinnings of regulatory strategies and decision making in biology, and that obviously necessitates the contribution of many fields and many institutions and many departments at UCSF are committed to that, including the QB3, the quantitative, the Institute for Quantitative Biology. He's at UCSF. That was, I think, number 10 on Mike's list of accomplishments. So let me start by actually defining something very obvious, but which is the topic of this session, which is what are the tasks that cells and organisms are faced with when they need to make decisions. First of all, they need to infer from incoming stimuli the probable state of the extracellular environment. Second, after sensing, they need to compute an appropriate response, perhaps through weighing advantages and disadvantages of all potential responses. And of course, they need to implement these decisions using evolutionary stable strategies that allow populations of cells either to outcompete their rivals or to survive environmental catastrophes. So here, I want to be clear. I'm not. I think a distinction that has to be made is that there's no implication about intent or consciousness of cells. Instead, the implications that we like to kind of live by are that cells have circuits. These are circuits or networks are implemented by biological entities that sense, compute, and actuate. And the same exact way, being an engineer, that I view our cars, the cruise control in our cars operate. The cruise control in your car senses the speed, computes the action, and sends the appropriate amount of gas to your engine to correct for any deviations from that speed. And this is kind of an analogy that and similar kind of analogies borrowed from the more physical sciences have been very helpful to us in our lab to try to get at these generalizable principles in biological organization. And actually, what it has taught us over the years is that in addition to formal operation in a digital way, we have to worry about also performance, about the subtle effects that lead to gain or lack of fitness in cells and organisms, and that have broad implications in terms of decision making. So although this strategy, this kind of analogy is actually very tempting and has we've gotten a lot of mileage thinking in these terms, I think what makes biology both fascinating and cryptic is that this analogy stops at some level in terms of biological properties. And let me dwell on these because I think these are going to be the frontier of research in the years to come. The first one of these is stochastic physics, and I think Rich just gave you a tremendous example about that, the role of stochastic physics in biology. So basically, in a nutshell, what I mean here is that at the very fundamental physical level, biology is implemented by collisions of molecules that diffuse in crowded cells, and these molecules and collisions happen with a certain probability. They don't happen deterministically. And this has many implications. One of which is cells, in the way they sense and compute, they are basically trying to solve a kind of probabilistic inference problem. And we know from engineering practice that actually solving a probabilistic inference problem has very different physical implementations than solving deterministic inference problems. So how can we factor that, knowing that the biological substrate is inherently stochastic? How do we factor that into our investigation of biological decision circuits? The other implication of that is actually more practical, which is this kind of stochasticity at the cellular level gives rise to heterogeneous populations. The level of heterogeneity or noise, as Rich called it, across this population is actually quantitative phenotype that has symptoms and of the underlying network organization. So a direction that we are pursuing really actively in my lab is how to actually exploit that information to expedite our biological discoveries. Another particular things about biological decision circuits is that they read the present, and sometimes they decide about the present course of action, but also about the future course of action. And let me give you a simple example of that. I think we all know about examples of that. But one example is, for example, in E. coli, when E. coli cells experience heat shock, the heat shock response is genes are upregulated, but also genes that are responsible for oxygen regulation. Or even though the heat shock shift has happened under plenty of oxygen. Although puzzling, when you put it into the right context, actually the explanation becomes rather obvious. E. coli cells, when they transfer from their environment into their host cells, they go through the gut, and the first thing they encounter is a heat shock, and then after that they encounter oxygen deprivation. So the oxygen upregulation is kind of an anticipatory response that puts the E. coli into its environmental and functional context. So the question then becomes, how do we factor this kind of anticipatory response that are actually also our window into the evolution of the organism, into our investigation of decision-making circuits? And perhaps something very related to that is what we call crosstalk, and this is the propensity of many cellular systems to actually use different cellular systems to use and reuse the same components. And traditionally we have considered that as a nuisance. How do you and we have investigated how different cellular responses that crosstalk with each other prevent this crosstalk to achieve specificity. But perhaps crosstalk is actually a way of cells to and organisms to decide about their future when they're computing in real time their present course of action. And of course, signal integration is a big factor that we need also to factor into our investigation. We in the lab we subject our cells and our organisms to one stimulus and we study one response. In realities, cells and organisms live in complex environment where they receive multiple stimuli, and their current regulatory organization and their decision making is actually always integrating these stimuli and prioritizing their response and so on. So perhaps we're at a time where we have to think about signal integration in a little bit more depth. And going back to a really important factor that Rich talked to us about in the context of biofilm is how do we think about individualistic behavior in cells and organisms versus community behavior? And biofilm is an example of community behavior. How do we think about bedhedging strategies and cheaters and cooperators in a community? And how do cellular decision circuits inside a single cell evolve to accommodate for community behavior? So let me leave you with basically, after this list of challenges and these complicated factors, you start being overwhelmed by the complexity of what we have to actually factor in our investigation of biological decision circuits. So how do we make sense of this complexity? How do we extract meaning out of it? And I would argue, and I hope this would set the ground for a lot of discussion afterwards, is I think we have to think about generalizable principles, about strategies and logical schemes that run across organisms. And here, this is where the value of multidisciplinary research comes in. We have to borrow from other disciplines where generalizable principles and extracting those have been the rule rather than the exception. And there are two ways, of course, in addition to borrowing from other disciplines so that we don't have to reinvent the wheel, there are two other areas where perhaps a lot of work can be immediately done. The first one of them is actually asking at some deep level whether there are preferred network architectures that accomplish any given biological functions. For example, a concrete example, is there a preferred strategies, irrespective of the physical implementation of it, that would be preferred to, for example, generate this population heterogeneity between chain and motile B subtle cell that Rich just told us about? Is it going to be a bistable switch all across, or is there another viable strategy that exists in nature to do so? The second direction that we can actually, with the advent of sequencing technologies and all of the modern era technology, it becomes more and more accessible and feasible, which is comparative studies across organisms. And here I want to go back to what Marc had mentioned in his introduction. I think we need to study model organisms and not only basically do human tissue studies, but maybe we should also think very hard about the principles that we really want to extract, and think hard about maybe new model organisms that will deliver the secrets of these generalizable design principles. And with that, I will stop and give the floor to Jonathan Weissman, who will tell us in depth or in a little bit more detail about some of the physical implementations of these strategies.
First, I really want to take the chance to thank Bruce for putting together this wonderful symposium. And of course, I'm honored to be given a chance to participate in it. So I came just a few years after Bruce left for the National Academy, but I've really benefited enormously from the environment that he played such a critical role in establishing, and have really enjoyed having Bruce back here and the energy and creativity he's brought to UCSF. Second, I'd like to thank Mike. We've heard a lot about Mike's accomplishments appropriately, and I'd like to focus just briefly on one, because it's the one that I've sort of most benefited from and most directly seen, which is, of course, the the move to Mission Bay and the creation of this wonderful campus. So I was one of the first labs to move. I think I was within about 4 weeks of the first lab landing at Mission Bay. And I have to say, I think like many of my colleagues, had a bit of skepticism about the move. And I guess I would sum up my feelings this way, which is if you're 90% happy where you are in your current situation, any change seems like it has a lot more chance for a downside than an upside. But really within a very short time of being here, it was clear that there are tremendous opportunities, and not only we'd recreated what we had at Parnassus, but that there was a terrific opportunity to do more. And QB3, which I'm most intimately involved in, is a great example of that. And of course, it's given me very concretely the opportunity to have terrific neighbors, Hao Li and Joe and Hana, who you've just heard from. So Hana, so now go into a little bit, comment on the science side. So Hana has rightly pointed out that there are design principles that cells have to adhere to as they try to solve their biological problems. And of course, it's important to understand and think about these design principles because they give us a context for studying biological systems and for understanding the solutions. But that said, biological cells have to find a physical implementation to solve the biological problem. And I think Rich's talk beautifully illustrated the incredible resourcefulness of cells in exploiting physical phenomena. In this case, and the topic close to my heart of use of amyloid fibers. Now, if a cell's going to communicate over a long distance, it essentially has to use a diffusible factor, typically a small molecule like deamino acids that we heard about. And that's a great way of communicating over a long distance, but these diffusible elements by their very nature are going to be evanescent. And if a cell, on the other hand, if a cell wants to communicate just to its nearest neighbor, it can rely on a physical contact. And that has an opportunity to be much more stable. But what about in this sort of mesostructure to communicate over multiple cells, but in a localized and stable manner? And here, the cells have exploited this unusual feature of some proteins to form these structures known as amyloid fibrils. So if you had picked up a protein biochemistry or structure textbook a few years ago, you would have learned that proteins can exist in two forms, either a soluble, unstructured form, the kind as a linear polypeptide, the kind of form that they're made right after they're synthesized by the ribosomes, or a globular structure, very well-defined structure, a little protein machine that lets it do its very specific function. This turns out to be a very incomplete understanding of the structures that proteins form, because we now know that most and perhaps the large majority of proteins can form under the right conditions these fibrous structures, often referred to as amyloids. And the key feature of these are cross beta sheet structures. So a strand of a beta sheet runs perpendicular to the long axis, and then they can go on indefinitely. So here's an electron micrograph of a typical amyloid fibril. And they're incredibly stable structures, but they're also self-propagating. And the reason they're self-propagating is by their nature they have an open end on the two ends of the fibril, and this can take an unconverted protein onto its end and catalyze this conversion. And this self-propagating nature of them has allowed them to form a new basis of protein or prion-based inheritance that, of course, was discovered here at UCSF by Stan Prusiner. In addition to this role in prion diseases like mad cow disease, they're also associated with a variety of other different neurodegenerative diseases, including much more common ones like Alzheimer's and Parkinson's disease. So there's naturally been a great interest, both from a practical and from a fundamental protein biochemical point of view, in amyloid fibrils. And as I said, the key feature of these fibrils is their self-propagating nature. That's illustrated here in this type of polymerization reaction, where one can take, in this case, an amyloid forming protein called sub-35, and dilute it from denaturant, and it will exist for an indefinite period of time in its random coil unfolded form. And then when induced to convert, in this case by a gentle rotation, it will rapidly polymerize into these fibers. that form these classic amyloid fibrils. Now, however, if one repeats this reaction and just puts a small amount of these preformed seeds in at the beginning, you don't need anything to induce the conversion, and the conversion it starts from the time 0 because these fibers act as a seed from which the unconverted protein can bind to the end and grow. And so this lets us in a test tube in a very simple system to replicate the core, to reproduce the core of this prion-based replication. And now, while amyloid or prion-based inheritance was, of course, discovered in the context of these terrible neurodegenerative diseases, we now know that they're exploited by normal biology. So just in the same way that, of course, DNA was first discovered or demonstrated in a pathogenic bacteria and in viruses, it's the basis of inheritance of all normal biology. So biology's exploited this amyloid forming ability. And the prototype of this came from in understanding the role of amyloids and prion inheritance in normal biology, came from the study of a rather obscure phenomenon known as Psi in the common baker's yeast, Saccharomyces cerevisiae. And Psi had a prion protein called sub-35, and sub-35 was more or less a garden variety protein. It had its role in telling ribosomes when to stop making proteins. But the unusual thing is it had appended, in addition to this normal function, appended onto a domain that came, as will, for reasons that will hopefully become obvious, a prion domain. And what this let cells do is exist in two states. One in which sub-35 was in its soluble, normal globular form, and this would tell ribosomes to stop appropriately. So if one had a reporter gene called AD1, for example, with a nonsense mutation, a stop mutation in the middle of the gene, AD1 wouldn't get produced and cells would be red. But now, and this would go on indefinitely, so this is a colony with millions of cells and every one of them is Psi minus. But now, if a fiber is introduced or forms de novo, there's a polymerization reaction forming these amyloid fibers through this N-terminal region, and this then allows it to propagate indefinitely through this self-replicating reaction that I demonstrated. It also does something else, which it depletes the normal sub-35 protein from the cell, and this depletion of the normal protein allows ribosomes to skip the stop signals in the middle of messages. So in this case, would allow AD1 to be produced, now causing the whole colony to be white. So in essence, what this lets the cell do is exist in two really fairly radically different states. This one seen here is the red for the Psi minus, and the one with the prion, the Psi plus. And to the nucleotide, the DNA is identical. You've heard a lot probably about epigenomes and methylation marks, et cetera, on DNA, but this has nothing to do with this change here. This is purely a protein-based change. And although Psi was the sort of first or one of the first normal prions, we now know that there's dozens of these prion-like phenomena in Saccharomyces cerevisiae alone, that the Psi phenomena is conserved through hundreds of million years of evolution, and that under some circumstances this ability to convert, at least in a test tube, seems to be confer an advantage to Psi. And so these are in simple model organisms, but we know that there's similarly looking prion domains in proteins that are in you and me and throughout eukaryotes. And in a really bold proposal by Eric Kandel, the Nobel laureate, he suggested that there's a specific protein involved in memory called CPEB that also might undergo a prion-like conversion, and that this prion-like conversion, in sort of the ultimate irony given the role of mad cow disease as a neurodegenerative disease, this prion-like conversion might be help cementing some of the memories, some of the physical basis of these memories. So that may be bizarre enough, but there's even one more bizarre twist, which is not only can a protein exist in two states, the soluble or this cell-propagating amyloid form, but the same protein can exist in multiple different conformations, each of these ones self-propagating. So that can be seen here, where you can take a soluble sub-35 and polymerize it either at 37 or say 4 degrees. We know these conformations are different by a number of detailed studies, and we know that these different conformations will seed themselves. So 37 seeds 37, 4 seeds 4. we can now introduce them into the same yeast, and if the yeast will then have a memory of the conformation that was introduced, leading to a clear difference in the phenotype that will be propagated indefinitely. So this then allows this decision not only to be prion or not to be a prion, but which flavor of prion to be, and sort of expands the repertoire of the possibilities of conformation or amyloid-based prion inheritance. So with that, I'll just end with a question that I guess we'll throw back to Rich, which is that it's clear that the key decision-making step in these types of conversion, whether it's an extracellular amyloid like we saw in B subtilis or an intracellular amyloid like the prion elements that I've talked about, a key step in this decision-making is when to convert and which form to convert in. And so then the question I would pose to Rich is, how is this decision made, and is this heterogeneity we see in Psi and so many of other prions also present in biofilms? So with that, I'll end.
It raises a question in my mind about this question of cheating, and about why wouldn't a B subtilis cell decide just to live off the biofilms made by other B subtilis cells. In sexual organisms which have germlines, these phenotypic cells, or you would say the specialized cells, don't contribute to the germline. So you're left with a cell which has almost no phenotype, which only succeeds by virtue of the community. But in this case, there seems to be ample opportunity for this sort of thing to happen. Do you have any thoughts about that?
Yeah, this is actually a fascinating question in general in microbiology, where there's multicellular interactions of various kinds. So in this case, I think there's an answer. So of course, you're right. A cell that mutated to be blocked in making the matrix would be at a short-term advantage because it's costly to make the matrix, and it could carry along with the rest of the cells. However, as you recall, the biofilm is temporary, and at the end of the day, the cells disperse and acquire an individualistic existence again, and then they go out on their own. And so the next time that particular cheater mutant cell by itself is stressed in a way that it wants to make a biofilm, it would be stuck. It would always be dependent on having other wild-type bacteria around. So I think it's this cycle back to an individual state and the wandering off on your own, that would be the Achilles heel of the cheater.
Good. Some other questions here from...
Well, Jonathan already asked me a question that I'm already been set up. I hope this is not going to be like a grill. So we know. All right. So Jonathan asked me, who's the nucleator for these fibers? And I by now I especially mean Roberto Coulter and his terrific postdoc Diego Romero have some clues. So it turns out there are two proteins that are needed to make these amyloid-like fibers. One is the principal component of the fiber. It's called TAZ-A, and the immunogold labeling was against that protein. But there's another protein. It doesn't matter what its name is. It's in the same operon. That protein is needed for the fibers to be made. It's located in the wall. We have genetic evidence that it's involved in sensing the amino acids, and there's biochemical evidence from the Coulter lab that it helps nucleate this amyloid transition. So we think we're on the right track, but there isn't nearly the detailed, beautiful biochemistry that Jonathan has done previously in yeast.
Hana, do you have a question, or do we have a question?
Are there any preferred topologies or network architectures?
Well,
could you think of something else that B subtilis could have implemented, and why didn't it?
Well, so specifically on this, maybe you're asking in the context of bistability, and So by the way, and I didn't use the word, but this is really epigenetics in the true meaning of the word. Am I going to get in trouble here? So this kind of memory through generations is what epigenetics means, and there are several ways to do that. And one is a double negative loop, as I illustrated. There's another conserved, recurring a motif for this, and that's a positive feedback loop. Now, it's not enough to have a positive feedback loop or a double negative loop. You also need cooperativity and non-linear response so that the two states can be stable. But in B. subtilis itself, and in other contexts and elsewhere, there are cells can exist in two very different states because if it's a positive feedback loop, it's off, or if the loop gets going, they're locked on. And so that's another example of memory, and I think that's widespread. In the persistence example that you were referring to from E. coli, there's another kind of phenomenon that's emerging that explains that, involving a threshold, and probably be more than I should go into here. But the two major answers are double negative and positive feedback, but not the only architectures.
Okay, so
Hi, how are you?
So, for single-cell organisms, and a lot we know a lot in bacteria, there's a real pressure for creating diversity, and the stochastic mechanisms that can switch to make populations heterogeneous have a number of advantages. And so these mechanisms have these features that Hana mentioned of stochasticity, but in a multicellular organism that's developing, these things have to be rigidly controlled, and when you follow the decision-making process there, it is remarkably invariant. And so the question is, how do you control these switches so that the switches are so well programmed?
But actually, there are some striking examples of stochasticity in higher cells. This is a wonderful story of Claude Desplan on the retina genes in the flies.
But it's a dramatic exception.
Okay. But it's dramatic. Yeah. Yes.
I mean, that's beautiful. So it is very beautiful. It's true, and it's a nice example. But
yes.
Its contrast to the normal situation is actually really startling, and that normally when you look at the progression in an embryo or in the nervous system, a specific cell is defined at a specific time in a specific place. With
let me challenge you with some more examples. I don't mean to mean dominant, but in odor, the receptor, Richard Axel's story. There's stochasticity in which cells express which. There's a huge number of odorant receptors in the mouse, and stochastically individual cells choose one or another. And the immune response, of course, that's
yes,
not.
But nonetheless, there are features. If you, I've done it most of my life now. Is to see if you look at the embryo developing and you watch the appearance of individual cells and you time their appearance, you decide what time is the cell going to divide. It divides at the same time in each embryo.
But Pat, isn't that just mostly true of things like Drosophila and C. elegans? I mean, if you look at other developmental systems, they're not quite so regular. The pattern of division is not so regular. Certainly there's a lot of regulation in development. You cut embryos in half, it goes back, and both halves will develop complete organisms. There are even some fish that undergo a lot of, the cells separate at some point in development and they dry out. These are these annual fish, and they make embryos with different numbers of cells, and they come together and generate things with not totally invariant situations. I think some of these organisms, which are evolved to be extremely efficient under very controlled conditions, tend to look invariant, but I'm not sure that's not a little of ascertainment bias. And,
Well, you remind me of years teaching developmental biology with you.
yeah, I was going to say, I was glad you asked the first question because, I mean, it also brought me back to, but anyways, I think that there is I think that, as you say, what you've said, I think is actually fundamentally true, but maybe it's exaggerated a little bit. The developmental systems really have constrained the behavior of cells, and how do you constrain behaviors of cells in these kinds of open systems? And I think that's certainly.
By the way, that's Patrick O'Farrell.
Yeah.
In case there happens to be someone who doesn't know.
Usually shy, but he managed to ask a question.
So actually, I want to clarify that I think we're all kind of saying the same thing here. If you start from the premise that biology at the physical layer, in a sense, is stochastic, and then you go to the behavior, and in bacterial community, you see some bacterial communities, you see the extreme where the stochasticity is perhaps amplified and exploited, and maybe in certain contexts of development, it's actually the other extreme where it's kind of attenuated. I think the interesting question here is how are both kind of scenarios implemented with the same with the same substrate, which is based on collision of molecules and so on. And I don't think we have a very deep understanding at this level of these aspects.
There were some other questions here.
It was just a comment.
Yes, go ahead. Comments are also.
I like comments.
So a comment on Patrick's response to Richard. And I think that, indeed, exactly what Hana was saying, it seems like we're talking about the same thing. What Richard is talking about is an environmental sensing, and all of the examples that you gave, Claude Desplan, etc., and olfaction, are sensors of a dynamic environment that changes, right? Whereas development cannot be that. It has to develop in the same way, irrespective of the environment. Indeed, the environment is structured such that information from the environment is reduced, the egg, et cetera, the nurse cells. And so what we're really looking at is exactly what Hana said. How does a biological system deal with the environment on one hand and being able to respond dynamically to odor, et cetera, versus being able to carry out its developmental plan. Anyway, sorry, that was just exactly what you said. Honestly.
One question over here, James.
Just in following up in this line of logic, I wanted to introduce another system that goes from a unicellular to multicellular state, which is dictyostelium. But what's interesting about dicty is that it forms actually a true community where some cells differentiate into spores or other forms. It sounds like what you're talking about, Richard, are not true communities, but really, in that sense at least, but communities of equals. One thing that's fascinating about dicty is that the variation of protein expression actually decreases in the multicellular form. They turn on an apoptotic pathway for certain cells. So I'd be curious as whether, just looking at, say, even something simple like the GFP protein expression levels, whether in a biofilm bacteria are less variable or more variable than they are in the individual state.
So actually, au contraire. So I oversimplified. In fact, there's in the biofilm, there are as time goes on, there are different categories of cells. And I was only talking about matrix-producing cells, but actually there are some swimmer cells, there are the matrix producers, and there eventually are some spore formers. And so it's something that has had a big impact on my thinking is for 35 years I thought about spore formation as taking place in individual cells, but actually in those biofilms, I didn't get into this, there were raised structures that look like primitive fruiting bodies with spores taking place at the tips, and with various double labeling with fluorescent reporters, it's a little bit like a tissue with different cell types, but not as sophisticated or as dictyostelium by any means, but not as homogeneous as you might have imagined.
Question, well, we'll come back to you.
I wanted to ask Jonathan, or Hana perhaps, among all of the prion assembly behaviors that you've seen in the yeast, are they irreversible or are they regulated and you can assemble and disassemble? And does that, if they can disassemble, does that give any insight to potential routes for therapy for prion disease in humans?
So
There's a question back there.
So the answer is they are reversible, as they would have to be, otherwise the entire population would convert. If once you convert, you can't go back. And they're reversible by changes in the environment that are physiologically reasonable. So high ethanol, for example, can induce prions to induce curing. And typically this is thought to act through the cell's chaperone machinery that act to start to disassemble these fibers or to prevent their propagation into the daughter cells. So there are not really to date a mechanism that would be sort of a capping mechanism, a physical capping of the amyloid, I think because it's so easy to break a new end off and to regenerate the end. But I think we certainly, society certainly been, and other prions have been a great model system for understanding how the cellular chaperone folding machinery, which you think of as being responsible for keeping proteins in their folded form, actually helps propagate these prion states.
There's a question back there.
So I think one of the questions that this brings up is how far are we from kind of understanding the cell as a computer? And is that even something that we can ever get to? Will it ever be even possible to take and integrate all of the inputs and come to a decision of where the cell will go? Or is that always something that will be completely impossible from a technical standpoint?
Hana, maybe you should handle that.
I actually, that's probably splitting hairs, but I would not call the cell a computer because in formal language, computing follows certain rules with the von Neumann computing rules and so on. And I do not think cells follow these rules. So I would be careful in calling cells computers or any analogies like that. So if I understand correctly your question, basically you want to give me the possibility to give me or anybody in this audience a set of inputs, and you want us to predict the exact internal state of the cell. Is that what we should be aiming for?
How close could you come? Like 90%? Or is it
Actually, unfortunately, right now, we don't even come close. And the reason for that is that we have a lot of parts lists. We know a lot of genes and proteins and so on. We don't know the logic. We don't know the algorithms with which they interact. And in a sense, this is the whole kind of point of a systems perspective, is to be able to kind of assemble these parts lists into perhaps quantitative models that will allow us to do so. We are still scratching the surface of this, so I don't know if we can do that in my lifetime.
Even just to put a fine point on it, what to me one of the great unsolved problems is how a protein recognizes a specific sequence in DNA. And we are very far from being able to predict that. So we can, empirically develop a weight matrix that describes it, but we can't predict it. So here's a little protein that can successfully find the right sites in the chromosome of the cell to bind to, and yet some of my very smart bioinformatics friends can't begin with a lot of neurons to predict how that happens.
Well, in terms of binding, I think Joe has some new method that would allow you to do so.
Well, I think that there's something you can do. First of all, I could, if the cell has two fates, I could get within 50% of guessing. So just there alone, I think there's progress. But in all seriousness, I think we don't want to set the impossible goal here. I mean, there's a lot of understanding, I think, that has come from rather simple models. Of course, the more quantitative information and the more complete the model, usually the better it is. And I don't think it's the sort of thing where we have to be able to solve everything globally, but if we can get some insight And I think each of us has contributed in little pieces of this along the way to models which have actually provided some real understanding, which without the quantitative analysis of this, or I should say without the formal analysis of this, with some quantitation added to it, it would just be a lot of hand waving. So in the spirit maybe of the last session, which ended with when are we going to eradicate malaria? Maybe we were going to, it's probably a little longer, but it's going to be a race. So when are we going to get to the point of these things being really deterministic? But I don't know, you want to say something?
Yeah, so I think in answering the question, it's critical to define the question in a way that's possible to answer. So we can't say where every protein molecule is in a cell, and it's sort of a fool's errand to try to do this anyway. And so the question is, what's the right level of abstraction? And as we've seen, when you have differentiation states, it sort of means something to be an epidermal cell versus a cardiac myocyte or some other type of cell. And those are complex states, but there's something similar about them that differentiates them from the rest. And also they're self-reinforcing, so trying to predict which of these states you're going to fall into once you get close enough that the self-reinforcing mechanisms will go in, I think is a much simpler and probably much more useful thing problem to try to solve.
Yeah, I think I guess in a way what Jonathan's saying is also that we don't necessarily want the extreme microscopic detail. We want to understand the important things, and that's the challenge. It's the art of all this. But I think we also have to have lunch. That's another important obligation of living organisms. So we'll see you later. Before you guys